sadly, we are leaving metabolism, so that might make you either really sad or happy. I love metabolism. It is, to me, it's one of the most interesting things to learn about because you really understand, right, what happens when you eat. So you can really relate to it. But I'm with you guys in which I understand that metabolism is kind of a beast. So, kind of sad, happy, right, for you guys. But now we're going to go into nucleotides and metabolism and replication, transcription, translation, all that. That's going to essentially be till the end of the semester where we're really focusing on nucleic acids. So this lecture is really going to be focused more on the very basic structure and function of nucleotides, and then the preceding lectures is really going to be more on really on their functions and their roles in the cell, such as transcription and replication. Again, this is really part 1. There's going to be a part 2 coming shortly. So this is really, again, going to be focused on introduction, and then part 2 is going to be focused on metabolism. So, so hopefully a lot of you have already taken Bio 120 or cell and molecular biology, and a lot of this should be review in a kind of a way. But what I really love about learning about nucleotides and nucleic acids in biochemistry is you really understand the chemistry of nucleotides, and I think that's pretty cool. Okay, so nucleotides have lots of different roles in our body. They are the precursors of RNA, so those are ribonucleotides, and DNA, those are deoxyribonucleotides, right? This is really the genetic material in our body. They're going to be essential carriers of chemical energy. So we know that ATP is very important for powering different chemical reactions, but we've seen different roles for other nucleotides such as GTP, UTP, and CTP. We've seen that nucleotides have very important roles in activating different types of biosynthetic intermediates. So if you guys recall, UDP glucose was really important for glycogen metabolism, so activating glucose in glycogen metabolism. Remember that in making phosphoacylglycerols, there was an intermediate called CDP diacylglycerol that was made, and that was again helping to activate the molecule for a downstream reaction. And we've also seen that nucleotides have roles in that they are components of different cofactors or coenzymes. So adenine is actually present in these cofactors that we've spent a lot of time talking about, such as NAD, so nicotinamide adenine dinucleotide, FAD, and coenzyme A. And some nucleotides, they act as cellular messengers. So we spent a lot of time talking about cyclic AMP that acts as a secondary messenger for activating protein kinase A, as well as cyclic GMP. We haven't talked, I don't think we really talked about cGMP, but it is a cellular messenger. Okay, so this is sort of, so shown here are the basic structures of the different nucleotides. There's adenine, guanine, uracil, and cytosine. So there's nucleotides and nucleosides. A nucleotide consists of the nitrogenous base. So it's going to be these either 6 and 5-membered ring or a 6-membered ring base that's then attached to a ribose sugar molecule. If it's in DNA, it's actually deoxyribose. I'll describe what I mean by deoxyribose in the next couple of slides. And then the ribose is attached to a phosphate group. So a nucleotide contains a base, ribose, and a phosphate group. You can have 1 to 3 phosphates attached. A nucleoside is just the base attached to the ribose. So there's no phosphate that's attached. So that's what makes a nucleoside. Okay, so nucleotides, you have the ribose sugar ring. So this is a 5-membered ring. It's this 5-membered ring here. And the carbon numbers within the ring structure, they are given prime designation. So here's ribose. This carbon is 1', 2', 3', 4', and 5'. And we give them those prime designations to distinguish the numbering of the atoms of the nitrogenous bases. So the nitrogenous bases, either pyrimidines and purines, they are also numbered from 1 to 6 or 1 to 9. And so they are not giving that prime numbering, so that way we know that, you know, if we're talking about carbon 1, carbon 1 prime, and we know we're really talking about an atom within the ribose and not the base. Now, a ribonucleotide is when you have a nucleotide and it has an OH group, the hydroxyl group, that is attached to the 2' carbon of the ribose ring. A ribonucleotide is when the OH group on that 2' carbon is replaced with a hydrogen. So instead of a hydroxyl group there, instead you have a hydrogen, and that makes it a deoxyribonucleotide. Okay, so the base is going to be either a purine or a pyrimidine. So base, the purine base is a 6-membered ring that's attached to a 5-membered ring. And then a pyrimidine is a 6-membered ring. And so these are heterocyclic and planar, meaning that you have different types of atoms within the ring structure, so carbons and nitrogens, and they're planar, meaning that they're actually flat molecules. And then in terms of the attachment of the base to the ribose or deoxyribose, it's joined covalently through what's called an N-beta glycosyl bond. Okay, what that means is that there is the N represents the nitrogen, either N1 or N9, that's attached to the sugar. Okay, so that's where the glycosyl comes from, in a beta conformation. Okay, that means that it's on the same side as the exocyclic carbon, which is the 5' carbon of the ribose ring. And so the N1 of pyrimidine or the N9 of purine is going to be attached through this N-beta glycosyl bond to the 1' carbon of the pentose ring, or of the ribo or deoxyribonucleotide. And then for the nucleotides, you can have phosphates, either 1, 2, or 3, so that's mono, di, or tri. And the phosphates, they're going to be attached to the 5' carbon of the ribose through a phosphoester bond. So essentially a molecule of water is removed between the ribose and the phosphate to make that phosphoester bond. Okay, so in terms of the compositions of purines and pyrimidines, both RNA and DNA, they contain purine and pyrimidine bases. Purines, these are your adenines and guanines, and so they are your 6-membered rings attached to the 5-membered rings. They are both in RNA and DNA. And adenine really kind of differs from guanine in that it has an amino group attached at this carbon, whereas in guanine you have a ketone attached at that carbon in guanine. Pyrimidines, these are going to be your 6-membered ring bases. They include cytosine, thymine, and uracil. And so cytosine is a pyrimidine that's in both DNA and RNA. And then thymine is in DNA. And then in RNA, that thymine is replaced with uracil. And so let's look at the structures of cytosine, thymine, and uracil. You can see that cytosine, it has an amino group attached at this carbon. With thymine, that amino group is replaced with a ketone, and it has this extra methyl group. With uracil, it looks just like thymine, except it doesn't have that methyl group in the, or attached to the ring. And so again, thymine is specific to DNA, and in RNA, you have uracil rather than thymine. And occasionally, thymine can be incorporated into RNA, or uracil can be incorporated into DNA, but that is really pretty rare. Just some fun facts. Apparently, guanine was first isolated from guano, bird manure or bat manure, and thymine was first isolated from thymus tissue, in case you ever wondered where did these names come from. I didn't know that until this year. And then in addition to sort of these standard common bases, there's actually unusual bases that exist that are found elsewhere, and they have different types of functional roles. You don't have to know these, but there's hypoxanthine, dihydrouracil. There's methylated forms of adenine and cytosine. Actually, cytosine methylation is a very, very common modification, and we'll talk about that later. And these can be found elsewhere, like for example in tRNA. Yes?
Does the methylation of the thymine specifically help something in DNA? That's why it's only exclusive to DNA? Or what's...
Yeah, so that's a great question. And I don't exactly, this is something I should really look up, but I don't know exactly where that specificity of thymine in DNA comes from, whether it might be DNA polymerase that specifically recognizes thymine over uracil. So this is something that I should probably look up, in the future. So but I'll get you that answer. I don't know. I don't know. Again, it does it, but I don't know exactly what lends that specificity. Great question, though. Any other questions? Okay. Okay, so anyways, this is sort of a table that goes over so the nomenclature of nucleotides and nucleosides and nucleic acids. These are sort of the technical names for the nucleosides and the nucleotides. If the bases are, in the nucleic acid, it'd be RNA or DNA. Typically, we just call them by their bases: adenine, guanine, cytosine, and uracil. And so, if I really call it so a lot of times I might be referring a nucleotide as the base adenine, or the base adenine if it's in DNA or RNA. So pretty much I just use the name of the base, and it's kind of just commonly used to describe whatever it is I'm talking about. Okay, so why don't we do a quick little exercise. Knowing what you now know about the structure of nucleotides, why don't you guys take a second and draw the structure of DADT. So that's deoxy adenosine diphosphate. you guys about 10 more seconds to try to figure it out. Maybe we'll use different markers to color coordinate today. Oh, I only have blue and green, so never mind. I'll alternate blue and green colors. Okay. So, first of all, I don't expect you guys to memorize the structures of the bases. I'm not going to lie. When I say memorize, in terms of being able to draw the structures from scratch, okay? I'm not going to lie. I can't even draw the structures from scratch. What I do want you guys to be able to do is be able to recognize the different structures of the bases. So if you have just, if I have on the exam or a quiz, for example, I'll have the structures of cytosine, thymine, uracil, adenine, and guanine, but I won't have the names under them. So you'll have to be able to tell, based on the structure, which base is which, if I were to ask questions about it. So, so I know that, let me just realize, missing our amino group here. Let me make sure everything else is correctly drawn. Okay, so remember adenine, it's different from guanine because it's got the amino group versus the ketone group. And this is a purine, so the numbering is 1, 2, 3, 4, 5, 6, 7, 8, 9. And so the base is going to be attached to the ribose via a beta N glycosidic bond. So I'm going to draw my ribose. So here's my ribose. I'm just going to leave it like this is. In a second, we'll kind of change it the way it's supposed to look like. So we want to draw the nitrogen, so that's atom number 9, attached to the ribose, right, at carbon 1 of the ribose in the pentose ring. So we're going to draw our N-glycosidic bond. So we're going to get rid of that hydrogen. And we're going to make it attached to carbon number 1. So here's carbon number 1. So here's 1 prime. And then here's 2 prime, 3 prime, 4 prime. And if we remember our the way that ribose is structured from learning about carbohydrates, that 5 prime carbon is outside the ring. Okay? And so this is our beta N glycosidyl bond here. So right, it's in the beta position, it's attached as a nitrogen, attached to a sugar, and it's attached from atom 9 in the purine to the 1 prime carbon in the ribose. And then in terms of attachment of the phosphate groups, it's going to be attached to the 5 prime carbon via a phosphoester bond. So here's one phosphate group that's going to be attached to another phosphate group. Now, is this structure correctly drawn? No. No. What's wrong with this? What was that? Right. So remember that I asked you to draw D, A, D, P. So it's deoxyribose, right? So that means that the way this is drawn, this is shown as a ribose ring, and remember that deoxyribose is actually lacking that OH group at that 2' carbon, so we should really substitute the hydroxyl group for a hydrogen instead. So good job, guys. So there's a pretty good chance that on an exam I would have you draw the structure of a nucleotide or a nucleoside. Yes, Mitch.
Just a quick question about the numbering.
Uh-huh.
So for the, if you're reading up there for the ring,
Uh-huh.
it starts with the 1 at what's the 3?
Oh, right, okay, yes. I actually had that number wrong. So it's actually yes. Good catch. So it's actually 1, 2, 3, 4, 5, 6. Apologize. All right, thank you. Okay. All right, anything else? So that was the other thing that was wrong with the structure. Okay, so now let's talk about how polymers are made from nucleotides. So DNA and RNA, that's the genetic polymer of information that is in our bodies, and it's really formed through the covalent linkage of the different of nucleotides. So phosphodiester bonds, they actually link nucleotides in nucleic acids, and this represents the covalent backbone of DNA. And so to make a phosphodiester bond, the 5' phosphate of a nucleotide unit is joined to the 3' hydroxyl group of the next nucleotide. And this involves the removal of water. So here is 2 nucleotides next to each other. And so here is 1 nucleotide. Here's your 5', or here's your phosphate that's attached to the 5' carbon. And then here's another nucleotide. You have the OH group that's attached to the 3' carbon of the ribose ring. And I'm actually just going to draw the phosphate group with an extra hydrogen on there, just so that way you can visualize how that water molecule is removed. So to make our phosphodiester bond, a molecule of water is removed. So water is lost. And then we form a bond between the oxygen that's attached to the 3' carbon to the phosphate of the phosphate group that's attached to the 5' carbon. And then this is what creates it's called a 3,5 phosphodiester bond. And so the phosphodiester bond is really between, so all this is the phosphodiester bond, and it's really this whole group, or the bond between the 3' carbon and the 5' carbon that is attached by this phosphodiester bond. Okay, so you have these phosphodiester linkages. They are always sort of repeating in the same orientation along a chain. And the nucleic acid strand has a very specific polarity. So a strand of DNA or RNA has distinct 5' ends and 3' ends. And so these 5' ends and 3' ends, they represent essentially the terminal ends of DNA and RNA. Okay, and I realize this is a little bit different compared to your notes. So so you have your 5' ends and your 3' ends. They represent the terminal ends of DNA and RNA, just like in proteins, right? Proteins have terminal ends. You have your N-terminus. The N-terminus has a free amino group on it. The C-terminus has a free carboxyl group on it, right? That's for proteins. In DNA, instead, you have these 5' ends and 3' ends. And so the 5' end has a free phosphate on it, and it's attached to the 5' carbon. I'm gonna turn off the light because it's kind of hard to see. So you have your 5' end, where the phosphate group, you have a free phosphate that's attached to the 5' carbon, and then the 3' end here, it has a free hydroxyl group on the end. So that hydroxyl group is attached to the 3' carbon. So this is a single strand of DNA. Here's your 5' end and 3' end, and here's your single strand of RNA. And so here's your 5' end and 3' end. Okay, so DNA, RNA has a backbone structure, very similar to protein has a backbone structure, and so it's really this repeating, alternating units of phosphate, or that's covalently bonded to either ribose or deoxyribose through these phosphodiester bonds. So this sort of part of the molecule here, so these repeating units of ribose and phosphates that are attached through phosphoester linkages, that's the DNA RNA backbone. That part of the molecule is highly polar. It's highly hydrophobic. Why is that? Sorry, it's highly hydrophilic. Why is that? Yeah, right, because phosphate has a charge, right? It's negatively charged, and also the ribose ring it's polar, right? Because you have an oxygen in the ribose ring, or same thing with deoxyribose. And actually, ribose is a little bit more polar compared to deoxyribose. So the hydroxyl groups of the sugar residues, they can form hydrogen bonds with water. So that's going to be more so in RNA versus DNA because it has that 2' hydroxyl group in the ribose ring. And the phosphate groups, they're negatively charged, and so they can interact through ionic interactions where the negative charge can interact with positive charge with other proteins or metals or other types of polyamines. The nitrogenous bases, you can think of nitrogenous bases as sort of like the side groups, right? So we think of, the proteins or amino acids in proteins, they have the side chains. So you can think of nitrogenous bases as the side groups in DNA. So they're joined to the backbone, that is specifically really they're attached to the ribose at regular intervals. The bases, they are hydrophobic. They are relatively insoluble in water. And they tend to be protected from water. And that protection is really coming through the structure, or the structure that RNA and DNA can form. And we'll talk about that in a second. Okay, so let's talk about directionality. DNA is normally written from 5' end at the left and then 3' end at the right. So if you were writing actually we'll do an example of that in a second. So it's always written from 5' to 3' direction. And then the phosphodiester bonds, they actually run in the opposite direction of the directionality of DNA. So the bonds actually run from 3' to 5'. So, looking at this DNA sequence and the RNA sequence, what is the sequence of DNA and RNA? One letter sequence. Yeah, right? So we're going to read this from 5' to 3'. So we're looking at DNA. It's ATG. So we read this kind of like we normally read, a book. That's from left to right. So for our DNA molecule, it's ATG. And then from our RNA molecule, we're also reading it from 5' to 3'. And so that's UGC. Pretty straightforward. Pretty straightforward until we only have the phosphodiester bonds to look at for information, which you'll get in the problem set coming soon. Okay, so DNA polymers, if you have a short polymer of nucleic acid, it's referred to as an oligonucleotide. So if it contains 50 or fewer nucleotides, we call it an oligonucleotide. If it's longer than 50 bases, then we call it a polynucleotide. Okay, so let's talk about DNA as genetic material. So, there's a very interesting and rich history by which, the structure and function of DNA was discovered. And this is all kind of relatively new information. So it's really kind of in the 1950s that all of these different experiments came about, and we really began to understand the molecular structure and function of DNA. So we're going to go over some of the interesting experiments that were performed. Okay, so one of the experiments that led scientists to conclude that DNA is genetic material and not protein are these sets of experiments that was performed by Avery, McLeod, and McCarty in the 1950s. So they essentially performed these experiments where they basically took either virulent or non-virulent bacteria. They did some different things with them, and then they injected them into mice to see if they lived or died. And that information helped them determine whether it was DNA or protein that is really the hereditary or genetic material in organisms. So in these experiments, they kind of did it sort of one by one, and and those different experiments allowed them to make their conclusions. So in the first experiment, they took a live encapsulated virulent bacteria, so it's deadly, and they took that bacteria and they injected it into mouse, so it died. So it told them that there's something in this live encapsulated virulent bacteria that was causing the mouse to die, and they didn't know if at that point whether it was DNA or protein. So then they did another experiment where they took that same bacteria, then they heat killed that virulent bacteria, injected it into the mouse, and it lived. So the heat destroyed it destroyed the virulence of the bacteria. Okay, so then they did another experiment where they took a different bacteria. It's a live, non-encapsulated bacteria, and it's non-virulent, and then they injected it into the mouse, and it lived. Okay, and then the next two experiments are the really key experiments that told them that DNA is genetic material and not protein. They took some of this heat-killed virulent bacteria, and then they mixed it with the live, non-encapsulated, non-virulent bacteria, and then they injected the non-virulent, or this mixture, into the mouse, and the mouse died. Okay, so what does that tell you? Yes, Mitch.
Conjugation going on?
Right. Well, close. Right. So there's something. You're on the right path. Right. Okay. There's something coming from the dead bacteria that was virulent. It was somehow transferring something into the non-virulent bacteria, right, and making them virulent, right? So it was transforming the heated, something in that material bacteria was somehow transforming that non-virulent bacteria to become virulent, and when it's being injected into a mouse, it was killing them, right? So they knew that, right, there's got to be something in that virulent bacteria, whether it's DNA or protein, that can be transferred into the non-virulent bacteria that's causing the mouse to die. Okay, so then they did the final experiment where they actually isolated either the protein or the DNA from the heat-killed virulent bacteria. Okay, so they did separate isolations. They then incubated it with the non-virulent bacteria, and they saw that when DNA was incubated with the non-virulent bacteria, it actually transformed the non-virulent bacteria and transformed it to make it into becoming a virulent bacteria. So then when you inject this into a mouse, it died. And that did not happen when you did this with protein rather than the DNA. So what this experiment showed is that DNA, not protein, is the genetic material. The DNA was important, is a material that's responsible for transforming the non-virulent bacteria to the virulent bacteria. Okay, so another experiment that it's done in kind of a different way. So they had a different technique for doing this experiment. This was done by Hershey and Chase, and it basically led them to the same conclusion that DNA is a genetic material and not protein. Okay. So in this experiment, again, the experiment was to see whether DNA or protein is injected by a bacterial phage into a bacterial cell. So a bacteriophage is a virus that specifically infects bacterial cells. And this is the experiment that they did. They took some phage samples and they radiolabeled it with P32, phosphate 32. Okay, so they grew some phage and gave it some P32. They took another separate sample of the bacterial phage and they labeled it with S35. Okay, and so the phosphate in the bacteria, the radiolabeled P32, it got incorporated into DNA, and then the S35 got incorporated into the protein of the bacteria, or of the bacteriophage. Specifically, how do we know that the phosphate is really labeling the DNA and that the sulfur is labeling the protein? Right, so DNA has the phosphate coming from the the phosphate backbone, and then protein, it doesn't have phosphate. Instead, you can find sulfur through some of the amino acids that can be found in protein. What are those? Yeah, cysteine and methionine. Okay. So they took their P32 labeled bacteriophage or their S35 labeled bacteriophage. They then incubated them with bacterial cells, and the phage, they will then infect the bacterial cells, and then so they'll infect the bacterial cells. And so they took their infected bacterial cells, and then they did this special treatment where they blended them to sort of shear off the viral heads, and then they were able to separate out the bacterial cells by centrifugation, and then measured which portion of the centrifuge material is radioactive. Okay, and so either they actually looked at the cellular part, which contains the bacteria, or the sort of the viral head part. Okay, and so they saw that when they centrifuged and separated the viral head from the bacteria, for either the P32 labeled experiment or the S35 labeled experiment, they saw that the radioactivity for the P32 labeled experiment was in the bacteria, but the bacteriophage was not radioactive. Whereas in the S35 experiment, right, the bacteria was not radioactive, but the viral head was radioactive. So what does this tell us in terms of what's going on? Right. Well, right. So it's really the DNA from the virus that's entering the cell, right? And so that is the genetic material. So here in the P32 experiment, the bacteriophage actually emptied its DNA into the cell. Okay, so that's really the genetic material that it's being that it's infecting into the DNA, and then right after it's being emptied, then the viral head no longer has the DNA, and so that's why we see that radioactivity in the cell of the bacteria versus the DNA. So this is showing us that DNA, not protein, is really carrying the genetic information in terms of which molecule is the genetic material. Okay, so let's talk about nucleic. Actually, I'm going to skip this slide for now. We're going to go back to this slide, and we're going to talk about Irwin Chargraff's experiments. So Irwin did some experiments that essentially figured out the composition of DNA. So he found out that the base composition really changes from species to species. So for example, the DNA in humans is different from the DNA in a dog. But if you actually isolate the DNA from different tissues of the same species, they have the same composition. So the DNA in our eyes is the same as the DNA, in our ears. Okay. The base composition is stable, so it does not change with age, nutrition, and environment. So my DNA sequence that I had as a baby is probably very similar, for the most part, to my base sequence that I now have as an adult. And he came up with these rules. So these are called Chargraff's rules, which is that the percent adenosine, the percent adenine is equal to percent thymine. So the number of adenosines are equal to thymines. The percent guanosines are equal to cytosines. And then and that A plus G equals T plus C. That is, the sum of the purines are equal to the sum of the pyrimidines in DNA. And so these rules are important because they provide key information for determining the three-dimensional structure of DNA. Okay, so to further be able to understand the structural nature of DNA, there were these experiments that were done by Rosalind Franklin and her mentor Maurice Wilkins in the 1950s, where they were able to essentially use X-ray diffraction to analyze DNA fibers. And so this is what the X-ray diffraction pattern looked like of the DNA fiber. And they could tell by this data that the DNA had a helical structure, but they couldn't really account for these rules that Chargraph was observing, and they could not determine the structure of the DNA helix. Okay, so in 1953, Watson and Crick, they actually were able to come up with the accurate description of the DNA. So this is James Watson and Francis Crick. Again, they're sort of credited for coming up with this description. And they essentially used the findings by Chargraph, so those Chargraph's rules, as well as the X-ray diffraction data that was generated by Franklin to be able to build a three-dimensional model of the DNA, so this double helix model that ended up being the actual structure, an accurate structure of DNA. And there's actually well, I'll let you guys do your own reading, but it's thought that actually Watson and Crick, some think actually, stole that diffraction data from Rosalind Franklin. But anyways.
One of them actually admitted later on in his life, after they got the Nobel Prize for it and everything like that, it's just yeah, we saw their data, and then it came to us, it just all clicked into place after that. But it's not conclusion based at all that was from that we learned it.
Mm-hmm.
Sorry, I'm very passionate about these two men in not a good way.
Yeah. Well, and yeah, so we're gonna move on. But they were right. So essentially, they still were the ones that came up with the model, right? They came up with the accurate description. So that's why I say they didn't necessarily discover DNA, but they did come up with the accurate description. Okay, so let's talk about this Watson-Crick model for the structure of DNA. The structure is these two strands of DNA that are wound around each other on the same axis, and they form a right-handed double helix. Okay, now this may or may not make sense to you in terms of right-handed versus left-handed, but if you take your hand, either your right hand or your left hand, and you sort of turn it inwards, that turn that your right-handed makes is the same turn as what a right-handed helix looks like. Same thing with your left hand. If you take your left hand and sort of turn it inwards, that would be like the turn of a left-handed helix. I'm not going to make you guys try to figure out what's right-handed or left-handed by looking at the structure, because to be honest, sometimes it confuses me. Okay. So they propose that it's got a hydrophilic backbone, where that hydrophilic backbone is on the outside of the DNA, and it is facing water. So here's your back. So the backbone is kind of in the blue, and you can see it's sort of on the outside. And then the hydrophobic bases, they are essentially on the inside of the helix. So they're in gray. They are closely stacked against each other in the helix, and in terms of the orientation, they're actually perpendicular to the plane of the helix. Okay, so the bases of one helix, they interact with the base of the other helix via hydrogen bonding, and the pairing of the two strands, they actually create what's called a major groove and a minor groove on the surface of the helix. And essentially, it's just like it sounds like. The major groove is sort of this groove or space that you can kind of see. It's sort of this major groove in the helix. You can see that there's sort of this, I don't know, cut away in the DNA. That represents your major groove. And then your minor groove is going to be sort of the smaller groove or space that is in the helix. A few other things. There's approximately 10 bases of double-stranded DNA per turn. 10 bases per strand per turn. So this is a whole turn of the helix. And then one turn of a helix is 36 angstroms. And then the width is 20 angstroms in diameter. Yeah. Okay. So one of the important things that they discovered is this hydrogen bonding of the two strands that occur between the bases. So this hydrogen bonding pattern is called base pairing. And so in the base pairing, adenine specifically bonds to thymine or uracil. So here's our adenine that's bonding to thymine in the base pairing. Or guanine, so G, bonds to cytosine. Okay, so you can have your guanine base pairing to cytosine. And this hydrogen bonding that occurs, that allows for the correct distance between the different chains of the DNA. So that allows it to be approximately 11 angstroms between the two different chains. So in terms of the AT base pairs, you have 2 hydrogen bonds between the two bases. And then in the GC base pairs, you have 3 hydrogen bonds. And so, so the GC base pairing is actually more stable because you have 3 hydrogen bonds versus the 2 hydrogen bonds that you see in adenine and thymine. And here is an important point, is that you really need to understand the hydrogen bonding. So if I were to show you, the 2 different structures, you should be able to figure out where, of 2 different bases, you should be able to figure out where the hydrogen bonds take place. Okay, quick question. So let's take a look at the bonding pattern of adenine and thymine. And we see that there's 2 hydrogen bonding pairs. Why isn't there hydrogen bonding between this hydrogen and that oxygen? Why isn't that? Pat?
The hydrogen isn't bonded to it's bonded to carbon.
Right, so the hydrogen here is bonded to a carbon, right? So that is a nonpolar bond, and so you can't have hydrogen bonding of the hydrogen to the oxygen. In order for that hydrogen bonding to occur, this hydrogen needs to be bonded to a polar. It needs to be an atom that would create a polar bond. So, for example, oxygen, nitrogen, or fluorine. Okay. Okay, so more about the model. The DNA strands are anti-parallel. It says something about 5,3 phosphodiester bond. Cross that out. It shouldn't be there. This is someone else's lecture, and it was not correct. Okay, so the strands are anti-parallel. That means they are running in the opposite direction. So one strand is going from 5' to 3' in one direction, and the other strand is going from 5' to 3' in the opposite direction. There is complementarity of these 2 strands in the polynucleotide chains of the double helical DNA. What that means is that you always have a purine. So you have your 6-membered, your 6 and 5-membered ring that's base pairing with your pyrimidine, so that's going to be your 6-membered ring. So you kind of have like a big ring that base pairs with a small ring. So that's going to occur in combinations of G, C, and AT. And the double helix is held by 2 forces, so that's going to be the base stacking interaction of the bases being stacked upon each other. This is largely nonspecific, and it really makes the major contribution of the stability of the helix, as well as the hydrogen bonding between the complementary base pairs. Okay, so let's talk about the different forms of DNA that can occur. There is actually a lot of rotation that can, or considerable rotation in that sugar phosphate backbone. So you can have bending, stretching, you can have some local unpairing of the strands, that's possible. And there's a lot of significant deviation from what we know as sort of the standard Watson-Crick DNA structure. So the Watson-Crick DNA structure is the B form of DNA. And these different deviations, they are found in cellular DNA, and they may play important roles in DNA metabolism. Even though you have these different forms, they don't actually affect the general properties of being complementary, of the strands being antiparallel, and this requirement of the ATCG base pairing. So that still holds true even if the DNA is taking on a different form. Okay, so the A form, so this is the structure of the A form. It's not really found in DNA. It has been experimentally found in low water environments, but it's probably not in the body. However, RNA, when it's double-stranded, prefers this A form helix. So the A form helix takes on a more condensed structure. The B form here, this is sort of the most common and most stable form of DNA, so we call this the Watson-Crick structure. And then the Z form is interesting because the Z form has the left-handed rotation, unlike the A form and the B form, which has the right-handed rotation. And it's got a more elongated structure and is more slender. And so there is evidence that it does exist, and it's not exactly clear what role it has, but it looks like it might have some role in regulating gene expression. Yes?
So RNA's existence in the A form, does that account for its like instability? Because like I know DNA tends to be more stable than RNA. Is that?
A lot of the instability of RNA is because a lot of the times RNA is single-stranded. So now in terms of whether, certainly the A form is less stable than the B form. I don't know if that's necessarily in the cell what accounts for the instability of RNA, but I think it has more to do with the single-strandedness that often it takes on. Yes, Brandon.
Is there a prominent DNA structure found in cancer cells?
I don't know. I would think that it probably takes on the B form, but I guess it's quite possible that maybe it takes on a different form in cancer cells. That's a great question. You should find that out for us. Any other questions? All right. Okay, so now we're going to go back to this slide that was sort of earlier. So in terms of nucleic acid structure, right? In the case of protein structure, you can think of nucleic acid structure as sort of, these different levels of complexity where you have your primary, secondary structure, tertiary structure, just like protein. So the primary structure of nucleic acid is the covalent structure and the nucleotide sequence. So for example, right, remember in protein structure, the primary structure of a protein is really supported by the backbone of the protein, the backbone amide bonds, and then you have like the sequence in terms of the actual residues of the amino acid. Kind of the same thing for the primary structure of DNA and RNA. So you have the covalent structure of the phosphodiester backbone bound to the ribose ring, and then you have the nucleotide sequence. So if you have a sequence of bases, A, T, G, C, this would be considered the primary structure. The secondary structure is any sort of regular stable structure that is taken on by the nucleic acid, and it's really related to the orientation of the backbone or sort of the nature of the helix. So these A, B, and Z forms of DNA, that would be considered secondary structure. And then tertiary structure is when the nucleic acid makes kind of a more complex folding arrangement. So tRNAs, they have a pretty complex mechanism of folding that gives them a three-dimensional structure. You have supercoiling of DNA within chromosomes. In eukaryotic cells, it's chromatin. In bacteria, it's nucleoids that can also form sort of this higher order, more complex structure. And we'll talk about that in the next chapter that we discuss. Mitch.
So is genetic modification, is that part of tertiary structure? Would that be like quaternary structure?
An epigenetic modification. It depends. If for instance you had methylation of this, it's kind of its own thing that's beyond the sequence and specific structure of the DNA. Although I think if it was methylated, so cytosines often get methylated, it would certainly be a gray area because I think it would certainly change the structure of the DNA. So I would say that so epigenetic modifications is really going to contribute to the tertiary structure. Yeah. Good question. And we're going to get to that in great detail later. Okay, so certain DNA can adopt unusual structures. An inverted repeat is a base sequence that have sort of twofold symmetry over the DNA strand. So if you have this DNA strand, you can have inverted repeats can really be kind of, they can come in many forms. They can come in sort of repeats of DNA that are like exactly identical to each other, or they're mirror images of each other, or they're complementary to each other. So if you have an inverted repeat in which that strand of DNA has complementary sequences with intervening bases. So for example, here you have a piece of DNA and you have TGCGAT. You have some bases, and then you have some complementary sequence, ATCGCA. They can actually base pair with each other at the complementary sequence, and then the intervening bases are going to form sort of this hairpin structure. So, so this is sort of a hairpin structure that can arise from this type of inverted repeat. If you have two of these strands of these type of inverted repeats interacting with each other, this can cause what's called a cruciform structure, meaning it kind of forms a cross. There's other types of repeats. A palindrome is when you have an inverted repeat with no intervening strands next to each other. So that's called a palindrome, and these two sequences are complementary to each other. So you can see, well, in this specific one, it's not. So many times it's complementary in the two strands. In this case, this is actually the mirror image of each other. And the mirror repeats, they are not self-complementary, but they are involved in triple helices. So you have, right, these two sequences, and they sort of mirror each other. Okay, and then direct repeats are when you have sequences that they directly repeat each other, but they're not complementary, and they can have specific roles. Okay, so let's talk a little bit more about stranded DNA helices. So certain DNA sequences, they can actually form triple or tetrahelices, and this can happen when you have hydrogen bonding that occurs with more than one base. And so these non-Watson-Crick base pairings are called Hookston pairs. So for example, thymine actually has the potential to base pair with adenine and thymine. This is adenine can base pair with two thymines simultaneously. So here's adenine. It forms two hydrogen bonds with thymine as well as two hydrogen bonds with another thymine. Or guanine can hydrogen bond with a cytosine, with one cytosine, and then with another cytosine that is protonated. So if you can imagine that these different bases are coming from 3 strands of DNA, then that can allow for a triple helix to form. So this particular Hookston base pairing, or base, well, this base, not base pairing, but this pairing, this guanine with a protonated cytosine and another cytosine occurs at low pH, and that's because the nitrogen within one of the cytosine becomes protonated. Yes? Does that interfere with anything? You know, I don't know. I want to say, I don't know. I don't exactly know exactly where these triple helices form, like what the functional roles are. Do you know?
No, actually, I have another question.
Oh, yes.
So for the designation, it shows like makes like adenine makes a double bond with thymine, but then the other thymine, it's just a dot between them. So I'm just wondering like why it's.
Oh, great question. Okay, so you have a great point. So you'll notice that so you have either 2 dashes or a little dot. Okay, the 2 dashes represent the normal, like the normal base pairing that can occur, and then the dot represents kind of like the uncommon base pairing.
Okay.
Yeah. So like for here, you have, so guanine forming 3 hydrogen bondings with a cytosine. So that's kind of the normal Watson-Crick base pairing. And then the dot represents the Hookston pairing, that sort of atypical pairing that we see. Great question. Okay. Okay, and then I'm not ever going to ask you a question about the G quadruplex, but you can even have, it gets complicated, right? You can even have like 4, like a helix formed from like 4 strands of DNA. This arises from Hookston pairing where you had pairing of 4 different guanosines coming from 4 different strand helices, and this generates what's called a G quadruplex, and this is found apparently in telomeres and as well as the regulatory regions of DNA, and apparently for the most part their function is still unknown. Okay, I think this is a good time to stop, and we will finish the rest of this on Monday as well as Tuesday as well as go into DNA metabolism.